Today I'm going to be going over Epibiology Unit 2, and this unit is all about the cells. So today I'm going to be talking majorly about the two main types of cells, eukaryotic cells and prokaryotic cells. I'm also going to be going over the organelles and the cell wall and the cell membrane that are in eukaryotic cells. So the difference between eukaryotic cells and prokaryotic cells really boils down to three things. In a prokaryotic cell, they store the DNA in a nucleoid, while in the eukaryotic cell, we store them in the nucleus. Eukaryotics are also much larger than prokaryotics, and in prokaryotic cells, there are no organelles, while in eukaryotic cells, we have many organelles that do the functions for us. So we have these similarities between prokaryotic and eukaryotic cells. Both of them have a plasma membrane. Both of them have a cytoplasm, both of them have a ribosome, and both of them have DNA/chromosome. These are the main essential things for all types of cells. So the main type of cells I'm going to be talking about today are these six, and I'm going to go into more detail later on. So the first thing I'm going to be talking about is the nucleus and the nucleolus. So the function of the nucleus is that it stores all of our genetic information, or DNA, and it is also the site for mRNA synthesis. mRNA is the RNA that helps in our protein synthesis. So this is very important since proteins are extremely important in our function. So the mRNA is very important in making those proteins. So that's where it's going to be made. We also have the nuclear envelope, which is the outer shell that surrounds the nucleus, and it's filled with pores, which are holes that allows travel in and out. So if the mRNA is made of the nucleus, it will travel out with the pores. The nucleolus is also the site of another RNA synthesis. This time it's the rRNA, which means it says ribosome. So the nucleolus is where ribosomes are made. So this is just an image of what the nucleus looks like. You can see this is the nucleolus, and this is the nucleus. You can see the envelope right around here. There's little holes, there's little pores. So the vesicle is the next thing we're going to talk about. These are membrane-bound sacs that are part of the endomembrane system. So what we call the endomembrane system is really just the system that resides all of the different membranes in our cell, because all of the organelles have membranes around them. So almost all of them are considered part of the endomembrane system. So vesicles are part of that system, and they usually reside in all sorts of places. Usually you'll find some in the cytoplasm, you'll find some in the ER. What they do is that they transport proteins and other stuff around the cell. So they're basically the transport vesicles, right? And then they can reside anywhere in the cytoplasm. So the endoplasmic reticulum, there are two types that we know of. We know of the smooth ER and the rough ER. So the difference in their name tells us the difference between them. The smooth ER is smooth, as in it doesn't have any ribosomes bound to it, while the rough ER does have ribosomes bound to it, which is why it's called the rough ER compared to the smooth ER. So the function of the smooth ER is that it detoxifies. This mainly happens in the liver. It also stores calcium, and I'm going to be talking about the function of that calcium later on when we talk about signaling. We also have the synthesis of lipids and carbohydrates. So that's the main functions of the smooth ER. The rough ER is mainly doing protein synthesis, which is very important. So that's what the rough ER is mainly doing. And it's also considered the membrane factory. What we mean by that is the rough ER has a lot of membrane around it. And what happens is when it's transporting proteins, that vesicle is going in and out, and it's easily able to just break off a piece of vesicle. And then the vesicle will travel to somewhere else and morph into another membrane. So that's what it means. It means it just basically provides a lot of the membrane that our cell uses. So this is an image of the endoplasmic reticulum. We have the rough ER right here. We can see that there's little ribosomes attached to it. And this smooth ER. You can also see that the endoplasmic reticulum is right next to the nucleus. That way it can transport proteins really fast between those two. So the Golgi apparatus is where proteins go after the endoplasmic reticulum. So the endoplasmic reticulum will make the proteins, and then they'll send a vesicle with the proteins to the Golgi apparatus. And at the Golgi apparatus, they will receive, sort, and ship the proteins. What that means is that they will receive the proteins, of course. They will sort them out so that they can go to where they're supposed to go. So it's kind of like a UPS or USPS or any shipping faculty. They will take them to their specific places, and then they'll ship them out to their specific places. So proteins can be modified in this stage. Some Golgi apparatuses will attach tags to them, or they'll attach a carbohydrate to a protein, and then they'll be modified, and then they can be sent to their final destination. So this is what the Golgi apparatus looks like. Some people call this a stack of pancakes. So this is what it looks like basically. And then that's just a vesicle coming in and out of it. So a lysosome is also part of the endomembrane system, and it's filled with hydrolytic enzymes. What that means is that the hydrolytic enzymes can break down other macromolecules. So the function it does is it digests macromolecules. So when a macromolecule is broken or no longer needed in a cell, that's where the lysosome will come in. The lysosome will come in and destroy them. So this often happens when a macromolecule is either in excess or it's broken. Like it doesn't perform its function anymore. This often happens with proteins. Um, the enzymes work in a very acidic environment, which is what the lysosome provides it. The lysosome has an acidic center, and that allows the enzymes to thrive. And when it wants to eat something, it will undergo phagocytosis, where it just swarms around the protein that it wants to eat, and the protein will go inside of the lysosome sac, and the enzymes will eat it. So if a lysosome explodes or bursts, nothing really happens, even though it releases a lot of pH, obviously, but it's just one lysosome, so that doesn't really have that much of an effect. The cell can easily go back down to homeostasis. But if a mini lysosome bursts, it can be the cause of self-destruction or apoptosis. So sometimes when a cell wants to perform apoptosis, a lot of its lysosomes will explode, and that's one way that it can perform apoptosis. So this is what a lysosome looks like. The little red acne looking dots are the enzymes, and you have a membrane around it. So the vacuole are just really large vesicles. The vesicles are made for transport, right? But the vacuoles are actually made to stay. So they don't really move around that much. Of course, they're made from the ER or the Golgi, which are known as the membrane factory, as I mentioned earlier. Some protists have contractile vacuoles. What that means is there's some protists that don't have that big of a wall around it, which means that water can come in and out really easily. And when water can come in and out of a whole entire organism that easily, they really need to be able to pump that water out or pump that water in. So that's what the contractile vacuole does. It will pump water in or out to keep the organism at the same water level. Plants will have a central vacuole, and that just means that it's able to store a lot of water because plants need a lot of water, and they have that cell wall around it to contain that water. So they want the central vacuoles to be really, really full or rigid. So that's what their vacuoles are made for. So this is in a plant cell. They have a really large vacuole. Plants usually have the largest vacuoles because they need the most water. So I know most people talk about mitochondria and chloroplast when they talk about the uh, organelles, but I'm going to be talking about respiration and photosynthesis very shortly. So I'm going to wait until then to mention it, just so you guys don't forget that information, because oftentimes I find that once we get to the cellular respiration and photosynthesis unit, where we really need to know the mitochondria and chloroplast, a lot of us have already forgotten it. So I'm going to wait until then to explain.